All right, hello everyone. As Kathleen mentioned, I'm going to try and give a pretty general audience-friendly description of coronavirus vaccines and variants today. And it should take you, I would say, 35 or 40 minutes, and then I'm hoping there will be some discussion at the end. So I'm more than happy to answer any questions that people have. But just by means of introduction, I'm going to break this talk into two general sections today, each of which will take about half of the time. The first half of the time, I'm going to be discussing basically what is a vaccine and why should you get one for COVID-19. And then after that, I'm going to transition to describing what is a variant that we've been hearing so much about in the news lately, and how concerned should you be about these variants that we're hearing about for SARS-CoV-2. So I want to begin vaccine discussion by trying to give you a sense of what it is that an effective vaccine does. So essentially, what a vaccine does is it serves to educate the immune system about which pieces of the pathogen it should target in order to stop infection. And you've all probably heard a lot about antibodies discussed related to coronavirus vaccines. But to understand what a vaccine is really doing, I kind of need to introduce you to the concept of what an antigen is. And so I like to use the analogy of describing the virus as like this car that you see here, where the body of the car is basically protecting the driver, which would be, in this analogy, the viral genome, the SARS-CoV-2 genome, which has the genetic instructions for the virus. And so if the immune system needs to stop this car, one of the things that it's going to do is try and generate antibodies or some way to basically block the car from functioning. The problem is that the immune system or your body doesn't a priori know which are the pieces of the car that are most important to block, right? You could envision that the immune system would make antibodies that, in this analogy, would recognize the hood of the car or the roof of the car or the rear taillight of the car, because these are all foreign components. They're all parts of the virus. But the problem is that these antibodies aren't necessarily going to stop the car. They're not totally useless. It can serve as flags to tell the immune system that the virus is there and be useful in some context. But these are not going to be neutralizing antibodies. What neutralizing antibodies are going to be are those antibodies that, when they bind to the virus, they stop the virus in its tracks. So in this example, neutralizing antibodies would be antibodies, for example, that bind to the wheel or maybe bind to the ignition or something like that, right? So if those are jammed onto the car, well, now the car can no longer go. And so this is what we're trying to get the body to specifically make antibodies to, is to proteins that are part of the virus that are essential for that virus to be able to get into cells and start the replication cycle. It's important to note that because in some cases, the actual virus, when it's infecting your body, can have strategies for sort of slipping through immune barriers, for masking and hiding some of those parts of the virus that are really critical to block it. And so in that regard, vaccines can actually help generate a more robust and certainly a safer immune protection in some cases than even natural infection can. And so that's one of the benefits. Beyond antibodies, of course, those are not the only components of an immune system that a vaccine is trying to elicit. And those other pieces of the virus are not necessarily useless as signatures or signals to tell the immune system how to recognize and get rid of the virus. So in the car analogy, if we were to say, put out an all points bulletin saying that, there is a car that does not belong on this highway. It is not from California, and we know that it's got out of state license plates. How is somebody going to search for that car? Well, this is akin to how our immune system starts searching amongst all of our cells to figure out which cells have been invaded by something foreign. So all of the cells in our body will display signs essentially on their surface that is giving the immune system this set of information. And I like to think of these as like license plates that are conveying information about, what is inside that cell, what's the make and model of that cell basically. So in this example, an uninfected cell, which was shown here on the left, if we were looking for, cars that should be there, that's California plates, will display on its surface a bit of information like a license plate saying, hey, this is California plates, everything is okay here, I am not the foreign car that you're looking for. Meanwhile, other cells that might be infected with one type of virus or another type of virus are going to display different sets of information, different sort of signs on their cell surface. They're going to take bits of that virus and point it out to the immune system as saying, hey, we actually have information here that is not the information that we should have. And this is basically alerting the surveillance, which is the cells of your immune system, that those cells are infected. And so these might be bits. So the license plate in the car analogy is not a bit where if it's bound by antibody, it's going to stop the car, right? It's not going to neutralize it. But nonetheless, that license plate has really important information that can inform somebody about where that car comes from. Does it belong here? Is it a stolen car? Is it a car that needs to be stopped? And so the cells that read these signs are components of the T cell part of the immune system. So you've probably heard a fair amount about T cells as being important. In particular, what these T cells do is they see these signs. If they're signatures of an infected cell, they'll read them and then they know to kill that cell. So it's not just blocking the ability of the virus to move and to get into cells, which is what a lot of times those neutralizing antibodies are doing. But it's also taking these pieces of virus and showing them in a cell that is infected so that these killer T cells can then come and get rid of those cells where the virus has managed already to get in and slip past those barriers. So usually an effective vaccine will trigger multiple parts of the immune system, including antibody and T cells. Mostly what we're going to focus our discussion on today is antibody, because we know that plays a really critical role in protecting us against COVID-19, and because it happens to be something that's much easier to study in patients and individuals than T cell responses. So we just have more information about antibodies and their protective role. So we know that on the surface of the coronavirus that causes COVID-19 is this trimeric spike protein, which I'm sure you've seen many pictures of in the news, and is sort of depicted here as these yellow spikes that are protruding from the virus particle. We know that spike is the major target of neutralizing antibodies against this virus. That means that when the body makes antibodies against viral proteins, those antibodies that actually stop the virus from getting into cells are the ones that are made against spike, all right? And it's not just a single antibody that can be made against spike and neutralize it, and this is an important point, but in fact, your body can make many, many, many different types of antibodies, all of which bind the spike protein in slightly different locations. And this is important when we come back later on in the talk and discuss variants, because it will help us appreciate what level of concern we might have for variants and how variants might decrease things like vaccine efficacy. So we know that these neutralizing antibodies, when they're made against spike, have the capacity to block coronavirus infection. I just want to spend a couple of minutes telling you why that is the case. Why is spike so important, and what justifies it as the focus of pretty much all of the vaccine efforts for COVID-19? Well, spike is doing two really critical things for the coronavirus that allow it to get into cells. The first thing that it is doing is it is recognizing components that are displayed on the surface of our cells that will allow the virus to get in. And so you've heard that spike binds a receptor on the cell. And so what this means is that essentially you can think of spike is like a master key, and it is looking for a lock to insert into because the virus needs to find a way to get past this cell barrier so it can enter the cell and deposit its genome and start an infection. If a virus can't get into a cell, that virus is for all practical purposes dead, and it cannot start an infection. It cannot copy itself outside of a cell. So it needs to get into the cell, but these cells are locked. And so you can think of spike as basically the key that the virus has to get into the cells. So spike is what's going to happen is it's going to come down. It's going to find this key here is going to find this specific lock, which is called the ACE2 protein on the surface of our lung cells and many other cell types as well. And when it docks into, sort of inserts that key into the lock, this starts a series of events that will allow the virus to sort of open that door to get past the cell. But just inserting the key into the lock isn't enough to allow the virus to get inside. It's got to somehow turn the key, push the door open, and how do all of those things happen? So that first step of receptor binding is really critical because it's finding the lock and key. But there's a second thing that spike has to do as well. And this is basically pushing the door open. And so this second activity is what's called a fusion activity, because the virus is coated with a membrane. The surface of the cell is coated with a membrane, which is just basically a layer of sort of lipids or fat. And if those two membranes can be brought very close together and fused and mixed, then the virus has an entry portal. And so spike has this second activity that is part of this protein that allows that to happen. And that second activity, that fusion activity, is tucked up in this lower stalk-like portion of the protein so that it doesn't activate until after the key is in the lock. But once the key is in the lock, you'll see that there was this second thing that happened, right? There was this little pair of molecular scissors called a protease that came and snipped off the tip of the spike protein, basically allowing this second activity to turn on. And so what happens there is that the second activity of fusion turns on in the spike, it unfolds. This allows spike to bring the membrane of the cell surface very close to the membrane of the virus, creating a channel, basically opening the door of the cell so that the viral genome can go inside and initiate an infection. So these two really critical activities, inserting the key into the lock and then turning that key and pushing open the door of the cell membrane, are absolutely critical for the virus to even start an infection. And so if you can block either of those, you can effectively block the ability of your cells to get infected with the SARS-CoV-2. And so for that reason, pretty much all of the vaccines that have been made and are in the pipeline are designed to show the immune system the spike protein so that the immune system knows this is the antigen, this is the factor that we need to make antibodies against and killer T cells directed against in order to stop the infection. So there are lots of vaccines that have been made and are continuing to be made against COVID-19. And I think of them as falling into two general categories. The first category of vaccine are those in which basically what the vaccine is doing is it's delivering a set of instructions to our cells to tell them how to make spike protein so that that protein can be shown to the immune system. The second general category of vaccines is that instead of giving our cells the instructions, we're giving our cells basically ready-made protein that's delivered directly into the body, all right? The vaccines that have reached approval first fall into this first category of delivering a set of instructions to our own cells and having our own cells make that spike protein to show to the immune system. These are the messenger RNA vaccines like the Moderna vaccine and the adenovirus vectored vaccines like the Chadox vaccine made by Johnson and Johnson and Janssen. So I'll explain each of these sort of separately, but conceptually it's kind of a similar thing. So what the scientists do to generate these vaccines is they basically take the set of instructions to make spike. And in the case of the mRNA vaccine, that set of instructions is basically written in a molecule called messenger RNA. And just like its name implies, messenger RNA is a molecule that basically takes information that would be in a genome and puts it in a form that can be read by the protein making machine of the cell, the ribosome. So it conveys the message to the ribosome of what the genome wants to be synthesized into. So our protein making machine is, our ribosomes are found in the cytoplasm of our cells. So what the vaccine basically does is it just takes a little snippet of this information that tells the cell how to make spike and encapsulates it in a lipid envelope that can get into cells. That message then is read by your own cell's protein making machine to produce the spike protein, which is then exported from the cell and shown to the immune system so that the immune system can make antibodies directed against spike, which will be neutralizing and presumably also killer T cell responses. Conceptually similar are these adenovirus-based vaccines. They're a little bit different though, in that instead of the instructions being in the form of messenger RNA, the instructions are in the form of DNA. And so what they're doing is they're taking a safe virus, one that does not cause disease in humans, and sometimes they're changing that virus even more so that it can't even replicate in humans, depending on the vaccine. And within this virus, they're putting a set of, again, instructions for making the spike protein. But these instructions are in the form of DNA. So then this virus enters human cells when you're vaccinated. It has to get into the nucleus because that's where DNA is read and made into messenger RNA. But then conceptually very similar, a message is created that tells the cell how to make spike. Spike is produced and shown to the immune system. It's very important to note that for both of these types of vaccines, the mRNA vaccine and the adenovirus delivered DNA-based vaccine, in neither case are these instructions inserted into our own genome, nor do they change our genome in any way. So these are not, which has been sort of a misinformation that's been propagated on the internet. These instructions don't go into your own genome. They're just read by the protein making machine, and then eventually they fade away and are gone forever after a short period of time. So they don't stick with us. And then the second portion here for ready-made protein delivery, basically instead of asking the cells to make the spike protein, scientists will grow up the spike protein on their own in the lab. This is called, and purify it. This is called a subunit-based vaccine, and it's also in phase 3 trials and getting ready to be administered now. Alternatively, you can just grow up large amounts of the virus itself, the CoV-2 virus, and inactivate it using a chemical to make that virus unable to infect cells at all. So you're just basically delivering an inactivated, killed virus, which has still spike protein on the surface. So in both of these cases, the immune system sees the protein that's already. So it's basically conceptually the same. It's just under one set of conditions, we're giving our cells the instructions to make the protein itself. And under the other set of conditions, we're making the protein in the lab and then delivering that protein to our body to be seen by the immune system. Okay, so I want to spend a couple of minutes addressing the question of why were COVID-19 vaccines developed and approved so fast? You will likely have heard that vaccine making is a pretty slow process most of the time. So before this one, the fastest vaccine that was made was against the measles, mumps, rubella vaccine, it took about 4 years. The Ebola vaccine was also, around that time scale, 4 or 5 years. But many other vaccines took 10 years or decades to develop. And so there was a lot of sort of suspicion centered around this idea of, if it takes years and years and years to make a safe vaccine, is the vaccine that is being cranked out in less than a year, were safety corners cut? Basically, can we trust this vaccine that it was, how do we know that it's as safe as those other vaccines that took so much longer to produce? And the short answer to this is that no, corners were not cut safety-wise. And so these are very safe vaccines, but that indeed there were a number of factors that had to be aligned to allow these vaccines to be developed in the rapid way that they were developed and with a high degree of safety. So let's go through what those factors were. First, we already knew that coronaviruses were viruses of spillover concern. So there had been 2 previous spillover events from animals into humans of highly pathogenic coronaviruses, the first being the original SARS coronavirus back in 2002, which spread around the globe and had a pretty high lethality rate, much higher than COVID-19 does. And then second, a decade later, the Middle East respiratory virus, or MERS, was in a separate spillover event, an entirely different coronavirus that spilled over from camels into humans and was a very lethal infection. It just didn't transmit well into humans. So the fact that 2 times in the span of 10 years, coronaviruses that were quite lethal in humans were able to cross the species barrier from other animals and get into us, gave virologists and scientists around the world cause for a high degree of concern that coronaviruses were something that needed to be looked at. So because of this, there was extensive research that had been being done since 2002, and even before then, in fact, but much more intensely since 2002. So it's now been nearly 20 years. That research had already established the importance of viral spike protein in viral attachment and fusion, and knowing that this is the target of most neutralizing antibodies for these related coronaviruses. They had already, in these related coronaviruses, developed vaccine platforms like the adenovirus-based vaccine platform, and done studies in primates and animal models to give people a sense that, in fact, these vaccines and those strategies had a chance of being highly efficacious. So we had a background level of information that was very good for these viruses because of that. Second is that over the last decade or so, there has been a significant advancement in new technologies for vaccine development. RNA vaccines in particular. So there had never been a vaccine made with RNA before, like the Moderna vaccine, but that's not to say that the technology had not been being worked on. So it had been optimized in the lab for something like 15 years with the idea of using this as a strategy for vaccines, and it had been used for other types of treatments in humans before. So it was known that it could be safe, and there had been studies suggesting that it could work for vaccines. We just didn't have a chance basically to test it. And so really that technology development was finally coming to fruition right around the time that COVID-19 appeared. But it was something that had been worked on for 15 years and optimized in the lab. So it was not it was new, but it was not untested or unstudied. Third, there was a massive influx of funding for COVID-19 vaccine research where, to the tune of $24 billion approximately was given by the government for COVID-19 research. There's no vaccine in history that has had that kind of financial backing by the government. And what the government was doing was basically assuming the financial risk, saying we're going to give all of this money to labs and to pharmaceutical companies to study ways to make this vaccine and then to produce the vaccine, mass produce it and stockpile it even before we know it's going to work. That way, once the phase 1, 2, 3 clinical trials are done, if they have good news, we're not going to have to wait another 6 to 12 months to now go back and make all those doses before we can give them to people. We'll make all those doses in advance, with the risk that if those vaccines turn out not to be efficacious, not to protect, we will have lost billions of dollars, but that is an acceptable risk. All right. So the government put the money up front to speed up that process, the production process, and the research process. Fourth, this being a pandemic, there was worldwide collaboration between academic scientists, pharmaceutical companies, and government agencies to share their data, share their advances as soon as they were coming out, so each group could immediately build on the work that was being done by another group. And this is sort of unprecedented in modern medicine and science. And then finally, one of the things that takes so long for vaccine development is those phase 3 trials to determine how well a vaccine works to protect against an individual against natural infection. And the reason why phase 3 trials often take years and sometimes decades is you have to wait for those individuals to get naturally infected with the virus. So you have a group of placebo receiving people who just got, saline or something like that, and a group of people who got the vaccine, and these are blinded, so you don't know who's who. You send those people out to do what they do on their daily basis in the world, and then you just wait for them to get infected. And so if there's not a virus that is circulating really widely in the world, it can take years and years and years just for enough people to naturally come in contact with that virus and get infected, to have enough statistical power for the scientists to be able to look at the data and say, oh, we can clearly see that this vaccine is working or not working, and how well it is working to protect these individuals. The fact that we're in the middle of a pandemic and the coronavirus is swirling around the entire world and infecting everybody at a remarkable rate means that that waiting period for how long is it going to take all of these vaccinated and placebo vaccinated individuals to come in contact with the virus, we didn't have to wait years. Those data, you only had to wait weeks. So this would very rarely be the case outside of a pandemic, because you just have to wait forever. And so that really sped things up. So it's this collection of things that allowed researchers and companies to basically advance vaccine development at a dramatically increased pace, not cutting safety corners, but taking the parts of vaccine development that usually take a while, that people usually do in a stepwise sequential way, like making tons of doses of vaccine, which normally a company would never do until they knew the vaccine worked, but being able to overlay that at the same time as you're testing vaccine efficacy really allows the process to be sped up. And so that was what contributed to the remarkable speed with which these vaccines came out. And the success, the efficacy, how well these vaccines work, I can say basically exceeded the expectations of pretty much everyone. We were all hoping for something that would be approximately 50% or more efficacious. And the fact that these are, in many cases, 90+% efficacious is just mind-boggling. Like, these are phenomenal vaccines. It is very rare to have vaccines out of the chute that have this degree of efficacy. And they've now been injected into 100+ million people around the world, so been very widely used. All right, I want to just address a couple of questions that are frequently discussed, which is, due to the speed of the vaccine rollout, what don't we know that might be known for other longer vaccine trials? So are there things that we should be worried about? Like, people say, well, you've only studied this vaccine for less than a year. maybe there are things you don't know about it that could put us, the general population, at risk. And what might those things be? There's a few things that I think are most relevant here. The first is we don't know how long immunity will last. And that's an important question. And so this is one of the big differences between this vaccine and other sort of FDA approved vaccines that are not under emergency use authorization, is that usually those phase 3 trials where you're testing how efficacious is a vaccine, well, they'll go on for years and years, right? You're often monitoring these people for 2 or more years. So by the time all the data come in, you have a good sense of not only does the vaccine work, but how many years out will it work? The problem for the COVID-19 pandemic, of course, is that you had situations where in the US, thousands of people were being killed a day due to COVID-19, particularly during the peak waves of these pandemic. And so we as scientists had data only for less than 1 year at that time. But what do you do when you have information that says, well, we know that from the 6-month time point we have at this particular juncture, that these individuals are dramatically protected, right? They knew that for at least 6 months, individuals who were vaccinated would be protected to a degree of 90+% against COVID-19 severe disease and death. Well, maybe, a year or 3 years out, that efficacy could wane to some extent, and the vaccine over time would become less good. But you can't wait that number of years to find that out while thousands of people are dying. You basically say, we don't know if it will continue to last for 1 or 2 or 5 years, but we know it lasts for at least 6 months. So we'd better get that out now and then continue to study as the pandemic goes on. And as time passes, we will get this information as it comes. All right, so that's something that those data will come at some point, but we just have to wait. You don't know if the vaccine is going to last 2 years until you're 2 years outside of the pandemic. And we're certainly not going to waste that vaccine opportunity before then. Second is we know that these vaccines protect against disease, which is the most important thing, right? We want to use a vaccine to protect against disease and death. That's what we care the most about, and they do that. But what's not known is to what extent they also protect against infection and transmission. So most vaccines actually protect against disease, but they don't necessarily prevent somebody from getting infected at all. So it's pretty uncommon actually to have a vaccine that completely protects you against getting infected at all. The measles vaccine is one of those sort of superstar vaccines, but many other efficacious vaccines don't necessarily do that. So this is something that is an ongoing question. Again, we will eventually know the answer. It just takes more time to collect those data, and we don't have a good sense of that yet, although early indications suggest that people probably are still getting infected once they're vaccinated. They're just not getting severely ill anymore. And then finally, sometimes people wonder about, well, how do we know if there's going to be long-term side effects that this vaccine might induce in us, given that it's only been studied for a relatively short period of time in humans? And while conceptually this might seem like a reasonable concern, as it turns out that there are no known side effects for any of the vaccines that are being used in the human population that didn't appear within the first 6 weeks post-vaccination, all right? Not to say that vaccines don't have side effects. Every medication has some side effect. Vaccines are, of course, our most safe medications that we have because there's a very low tolerance for side effects because you're giving them often to healthy people. But things like allergic reactions and other sorts of rare side effects that do pop up, they always have popped up within the first 6 weeks, and usually for allergies even before then, post-vaccination. So while in theory, it is possible that there might be some longer term side effect, there is no precedent for that ever having been the case post 6 weeks, which this vaccine was certainly studied for longer than 6 weeks out. So I think that's a concern that is not, should not be a major one. All right. I'm going to transition now to talking a little bit about variants. This is going to be somewhat shorter than the vaccine discussion because that also included the introduction and whatnot. But let me just start with a general way of thinking about variants with another analogy. And this time I want to make an analogy to a beehive. So a beehive, right? You've got a swarm of bees that are worker bees, and you've got a queen bee. And so I want you to think of the original coronavirus that started the pandemic, or sort of the consensus virus. That is the queen bee, all right? The queen bee is the source of all of the worker bees that are part of a hive. She produces them, so they're all very related to her, but they're all a little bit different, right? They all belong to the same hive, but they all are a little bit different. And so you can think of the queen as being like the original virus, and all of these worker bees as being the variants that happen to emerge as that queen bee is sort of mass producing more and more eggs and progeny. Occasionally a new queen bee will come, right, and start a new hive. You can think of that as a entirely new strain of virus, which starts a new hive. But these variants that are swarming around the queen bee virus basically are just small differences, usually compared to the main virus. So how do these differences arise? Well, when a virus replicates in a person, it's basically making millions to billions to sometimes trillions of copies of its set of genetic instructions, its genome. And when you're making that many copies of something, occasional copying errors are inevitable. I want you to think of just yourself as you are typing a paragraph. Let's say you had to type a paragraph over and over and over again. Even if you just had to type it 10 times, the statistical chances that you would make an error, even if you're a great typist, are pretty high, right? If you had to type it 100 times or 1,000 times or 5,000 times, the chances that occasionally you're going to make a typing error, and it's going to be a random error somewhere throughout that paragraph, are basically going to be there. Even if you have you've got your autocorrect on Word, autocorrect doesn't correct everything. There are some errors that it won't recognize and know how to autocorrect. So you're still going to have mistakes that are made as you're copying that paragraph. And that's the same for the virus. As it's copying its genome, occasional random mistakes are made, especially when you think about the staggering number of copies that are made. And so I'm going to just walk you through an example of what this might mean. So let's say in this analogy of you're typing a paragraph, that one of the sentences for that paragraph, the initial sentence that you were typing was, you've got something, I was eating crisps for lunch and they were an odd color. All right, this is a sentence that originated, let's say, in London. All right. And so as this individual here needs to type this paragraph over and over and over again 100 times, occasionally she might make a random error that basically isn't doing anything here or there for how this sentence is read. For example, maybe she accidentally adds a second space between, so there's two spaces between these two words instead of one space. That doesn't change the meaning of the sentence at all. It doesn't change the interpretation of the sentence at all, no matter who's reading it. Maybe an extra comma is added. These are basically variants that are neither here nor there for the virus, really, or for the meaning of this sentence. However, occasionally she may make an error that basically makes it extremely difficult to understand the sentence because it changes the meaning of it. So here, instead of crisps, she substitutes accidentally a B for a C. I was eating brisps for lunch and they were an odd color. Well, we're going to look at that and you're going to think, I have no idea what this person was eating for lunch. I don't know what brisps is. I don't know how to make an assumption as to what it is. I don't know how to hypothesize what it is. This is a detrimental error that has been made. It changes the meaning of it for the worse. So this would be a change that, if it happens in the virus, would basically cause that virus to wink out of existence again, because the virus basically, it is deleterious for the interpretation. Alternatively, occasionally you might make an error that makes the clarity a little bit better. For example, instead of crisps, she accidentally types, I was eating chips for lunch and they were an odd color. Well, in the US, we call potato chips. In the UK, they would call them potato crisps. So if this was being read in the United States, in fact, this sentence would be more easily understood regionally by people in the US, and this sentence would be more understood perhaps regionally in the UK. So this sentence would actually work better, slightly better under these particular conditions, depending on where the geographic location of the reader is. And then additionally, you could have changes that are built onto that, like instead of I was eating chips for lunch and they were an odd color, spelled in the British variation of it, she types odd color in the US version of how color might be typed. Well, this is interpreted the same, whether probably you're in England or you're in the US. But if you're having to type a sentence 100 times, 5,000 times, having one less letter allows that sentence to be typed a little bit quicker. So this will probably be adopted because it confers not only more clarity if this sentence is sort of circulating through the United States, but also makes it easier to copy it, be copied a little bit faster. So this analogy of a word change that may be more clear only in a certain geographic location actually applies to viral variants as well. So you can think of each of these as being variants of this original sentence and what might happen to the virus as it's copying itself. And the reason why these word changes in geographic locations and their meaning might be applicable here as well is that it's hard to reliably compare human study results from different geographic locations. So there's a lot of complications. People have different social habits. Maybe in some social groups, it's common to kiss somebody hello. Other people have much more social distance. What are their sort of gathering patterns, etc. Different nutritional status, depending on the location around the world where you are, different medical and public health infrastructure, different parasite burdens that will change our immune systems in subtle ways. And so a variant that behaves one way in one part of the world, it's not always easy to extrapolate and say it's going to behave exactly that way if the variant comes to another part of the world. So we're hearing a lot about these worrisome UK variants, South African variants, Brazilian variants, and we're assuming that the sort of behaviors that those variants have in those locations are going to be mimicked when those come to the United States. And that may be true, but it may not be true as well. Another complication is that we've heard that some of these variants can cause reduced vaccine efficacy, particularly some of these variants of concerns, but that's a little bit challenging to understand well at this stage because they're coming from studies with relatively small numbers of people, but are being compared to very large randomized phase 3 trial data for the vaccine. So whether or not the reduced efficacy that is seen with these small studies is truly reflective of a decrease in vaccine efficacy, I think is going to need to await more data. So these are important clues that scientists are getting and public health individuals are getting, and certainly warrant very close following. But I think it's hard often to make large assumptions about what a variant is going to do in terms of vaccination or transmission and increased transmission or disease, for example. So variants are worth watching really closely, though, not only because they could increase transmission. In particular, several of the mutations in the circulating variants of concern have changes in locations on spike that are bound by neutralizing antibodies. So remember, I told you that all of the vaccines are using this spike protein to generate antibodies and neutralizing antibodies against spike. Well, the variants of concern, and part of the reason why there are variants of concern is that when people look and see where these changes are, Some of those changes are in just locations of spike where we know that neutralizing antibodies are bound. So in these teal stars here, when you have a mutation in this location, well, that changes the bit of spike here so that it is no longer recognized well by this neutralizing antibody here. Or in this mutation, hypothetical mutation, this prevents effective recognition by this neutralizing antibody here. And so the reason for this is that as the virus is encountering what we would refer to as pressure from neutralizing antibodies that are present in the population because of vaccination and natural infection, these variants with mutations that can escape any of these antibodies are going to gain a slight advantage and start to predominate. And that's why we're starting to see some of these variants spread. But the good news is, as I told you in the beginning, the neutralizing antibodies that are made against spike are not just of one variety. So stopping this one or stopping that one doesn't mean that there are no neutralizing antibodies left. There are still going to be a suite of neutralizing antibodies that can, in theory, block spike. So it may be a little bit less protective because you've lost a couple of those sites, but it doesn't mean that all of a sudden the vaccine efficacy is going to go to zero. All right. So I'm just going to end now and say that the best strategy to prevent COVID-19 and the spread of variants while you're waiting for your chance to get your vaccine is still the same as it was even before we had the vaccine, which is wear a mask, wash your hands, and watch your distance, because preventing variants is no different. Preventing spread of variants is no different than preventing spread of that original virus. And so it's not that the variants can magically make it through the mask better or anything like that. So they can still be stopped in the same way through these same public health measures as the original virus can be stopped. So I will end there, and I am happy to take any questions if people have them.
I'm going to go ahead and stop the recording, and then we can do like a question and answer session.